or I hope.
Mhm.
So it's my pleasure to welcome Petr Kohout to talk about gradients, altitudinal and latitudinal. And it's a nice paper published in New Vital, and we are all ears hearing what you found and how, and let's discuss it thereafter. Thank you very much, Petr, for accepting.
Thank you, Honza. I would like to give you a brief intro to our relatively recent paper, but very old study, to be fair, which was published in a New Phytologist. And as you see, there are many co-authors. I would like to first thank everybody who were joining us in the field and in the lab to do this. From my point of view, very nice work. And what is the work about, or what is the background that we are building on? As many of you are not like very much ecologists, so I will give a bit background of our study. Our study was focused on altitudinal and latitudinal diversity gradient and what are the drivers of the diversity gradients in case of fungi that we can see. Why the altitudinal and latitudinal diversity gradients are interesting and why we think that this is important. This is going back to Alexander von Humboldt, who was the first biologist who discovered or recognized that mountains are like a small scale gradients similar to the latitudinal gradients that we see on the global scale. You probably know that the highest diversity of the organisms is situated in the tropics, where we have the tropical moist forests, especially in South America. And then it is the biota, vegetation, and consequently also the animals and everything else is changing the diversity composition and so on towards going along the latitudinal gradients northward or southward. So Alexander von Humboldt was the first one who recognized that the mountains are some kind of like a small latitudinal gradients, but along altitude. And this recognition went through, and then this recognition was used to understand many features, how diversity of the various organisms is structured, and what are different, let's say, environmental characteristics that are behind the structuring of the diversity patterns. Basically, we did the same. We used the beauty of the altitudinal gradients as some kind of natural laboratory to study environmental drivers and stochastic drivers on assembly rules of fungal communities in our case, and diversity patterns of the fungi. Most importantly, we focused on the environmental drivers that are behind structuring of fungal communities, like climate, because we are in ongoing climate change. So climate is typically the most studied and also the most influential environmental driver that is determining fungal species distribution, fungal richness distribution, and so on. And then productivity and others like soil chemistry, and also on stochastic drivers. And that's a bit more tricky to understand because stochasticity is, first of all, it's some random factors. In our case, we focused on stochastic driver that is primarily determined by, or primarily driven by geometry. By simple geometry, it is called mid-domain effect. And if you imagine altitudinal diversity gradient, and you would start fitting on the altitudinal gradient along some slope of the mountain, and you would start fitting individual species distributions just randomly. You would take randomly like a species that is present on here over here, and so on and so on, and you would just randomly put them on the altitudinal slope, very likely you would end up in high diversity in the middle side, in the mid, let's say, elevations, because just by random chance there will be the highest overlap of all the species distributions. So what we were trying to do, we were trying to disentangle actually effects of the deterministic and the stochastic factors on the distribution of fungi. And we particularly focused on 3 fungal ecological groups: saprotrophic fungi, mycorrhizal fungi, and fungal root endophytes, that were, let's say, the most prominent in our sampling, and also are very important ecological drivers of, for example, carbon cycling of nitrogen, phosphorus, and having mycorrhizal fungi are having tremendous effects on the vegetation. And root endophytes are, for example, important also in carbon sequestration and so on. So therefore, we were focusing on these 3 groups. And what we did, what we aimed, we assessed the gradients of diversity for these actually ecological guilds along the altitudinal and latitudinal diversity gradients. I will show them on the other slide. Then we identify underlying mechanisms behind diversity patterns, and we modeled species richness of the different fungal ecological groups along the elevation and latitude. After accounting the environmental variables, I will show you the results later. That's probably the most interesting result. And we aim to disentangle the architecture of the diversity patterns by verifying if the individual species occurrence reflect the general diversity patterns. How we did it? We did a relatively labor-intensive and time-intensive sample or time-demanding sampling in the whole Europe. Each of the dot here is representing one elevational slope in various mountain ranges all over Europe, and on each of these elevational slopes we sampled we sampled some elevational gradient on each of the sides. We had several samples of the soil, and in each of the sample we assessed the fungal diversity using metabarcoding methods. In our case, sequencing of the ITS ribosomal RNA. And what were the results? Unfortunately, most important are the patterns, and I will guide you through them. There will be too many pictures, and there will be relatively small, so sorry for that. First, I will show you the results of the general diversity patterns that we see in case of different fungi and different fungal groups along altitude and latitude. These are raw data without accounting for any environmental variables, for anything. These are the raw data that we see actually. And what we found out, that basically the fungal richness is changing way more along altitude than along latitude. And we see something what we would expect in case of fungi and saprotrophs, that the diversity of all fungi or saprotrophs themselves is decreasing along elevation. So more higher in the mountains you are going, there is a less fungal richness. And in case of ectomycorrhizal fungi, we saw something what we call this hump shape effect. We saw the highest diversity of ectomycorrhizal fungi in the mid elevations. So let's say they peaked actually in this let's say 1,500 meters above the sea level and so on, and they decreased towards lower elevations and higher elevations. In case of root endophytes, we did not observe any pattern in altitude along the altitudinal gradient, but we saw that the root endophytes are increasing, their richness is increasing along latitude, while the latitude was not significant for any other groups. So these are the raw data. This is some baseline of the data. And then we were interested, what are the environmental drivers that are driving or that are causing the patterns that we see. I will not go into details here. I just want to point out that you can see that majority of the drivers that we identified were somehow related to climate, while the soil chemistry variables did not have such a pronounced effect on the richness of the fungi. And now the complicated but probably the most interesting part of our study is these graphs, over this one over here and the first one over here, are the same as in the previous figure. So they are showing the raw data without taking into account any environmental variables and so on, showing the patterns of the diversity of these groups along altitude and along latitude. And the other two columns are showing residuals. Sorry, forget about residuals. Are showing the data of the fungal richness after taking into account the effect of environmental variables that are having on the species, on the fungal species richness. In other words, I can explain it, for example, on this one. If we account for the effect of climate in case of all fungi, we see that suddenly the pattern that we see, the decreasing diversity, disappears. So this is telling us that behind the decreasing diversity that we see here is the climate itself. Because when we exclude the effect of the climate on the fungal species richness, we do not see any pattern anymore in the data. So everything can be majority of the effect that we see can be attributed to the effect of climate on the distribution of fungi along the altitude. The same is true for the saprotrophs. The same is true for the increasing richness in case of root endophytes. When we take into account the climate, suddenly there is no pattern in the diversity. So everything can be attributed there. But there is one interesting group where the pattern remains even after accounting for the environmental characteristics. And these are ectomycorrhizal fungi. When we account for the effect of climate, they still show the hump shape effect. It's smaller, but it is still significant, and they still show the hump shape actually gradient of the diversity. And now, what is behind it? I was telling you in the beginning about the mid-domain effect. So what we can think, or what we might actually assume, is that the diversity of the ectomycorrhizal fungi is highest in the mid-elevations because there is an overlap of the distribution of individual fungal species in the mid-elevations. This is this mid-domain effect, that if we put together all over the fungal species distributions, we will find the peak of the diversity in the mid-elevations. And to test this, we modeled distributions of individual fungal species along the altitude, latitude for ectomycorrhizal, for saprotrophs. The saprotrophs are not that important for us right now. Let's focus on the ectomycorrhizal fungi and on the elevation, because this basically is explaining what might be behind this pattern. And then we expected two potential scenarios. We expected that if we see, or if the mid-domain effect, if this geometry is playing the role, we would see that half of the species will show decreasing distribution along the elevation, and the other half will show increasing increasing abundance along the elevation. But we didn't see any of that. We saw exactly the opposite. We saw that that majority of the species that somehow responded to elevation showed again this mid-domain effect, which is telling us that ectomycorrhizal fungi are showing highest richness in the mid-elevations because majority of the species find the optimal environmental conditions there in the mid-elevations. It's not driven by the geometry, but it's driven by the environment, but not by climate or the soil chemistry. These were taken into account. So we have to find something else. We had to find some other explanation. And the explanation is the last sentence over here. The lower ectomycorrhizal fungal diversity in low elevation is driven by the specificity of local environmental conditions, as low elevation forests are more prone to current and historical anthropogenic disturbances. In other words, if there was no human activity, if there was no human pressure, we expect that the ectomycorrhizal fungi would show decreasing richness along the elevation, but we think that these lower elevational forests that are under high environmental or anthropogenic pressures have, let's say, decreased diversity, and therefore we see this hump shape. So this hump shape distribution of ectomycorrhizal fungal richness along elevation is not driven by climate, is not driven by soil, is not driven by stochastic effects, geometry, and so on. It is rather the effect of environmental pressure that humans and our civilization is affecting these lower elevational sites. And we see very similar patterns. We, for example, know from Kilimanjaro, from Kenya, that butterflies are showing very similar pattern when there is a high pressure of humans on the lower elevations, and they are also showing this mid-domain effect that is unexplainable by other factors than basically the human activity. Besides this, just to conclude, we showed that the environment differentially shapes the diversity of fungal ecological groups along elevational and latitudinal gradients, and however, this is not the true for the ectomycorrhizal fungi. I would like to point out that our paper was also, or is receiving a relatively lot of attention, I would say, because new phytologists provoked Peter Kennedy and Matthew Smith, who were most likely reviewers of our paper, to write commentary on our paper, which is not, I wouldn't say it's some trashy commentary. It's not like trashing our paper. It's rather to point out that it is relatively interesting. And they are summarizing that this work provides a much needed perspective of whether mountains function as a latitudinal analogs for soil fungal communities and how different fungal lifestyle mediate biogeographical patterns, and that our findings somehow confirm and challenge our prevailing understanding, particularly regarding the universality of the mid-domain effect as an explanation for fungal evolution, elevational gradients. So yeah, this is all. I would like to thank many people from the lab because it was a work of really many of us. And thank you for your attention and for coming here. Thanks.